Hi, this is Dr. Tracy Zhang, and I'd like to talk to you today about neuron anatomy and action potentials. Our objectives today are to discuss the types and anatomy of nerves and neurons. We'll talk about how neurons communicate via the action potential. We'll look at parts of the action potential, including depolarization, action potential, repolarization, refractory period, and resting potential. To begin with, we're going to talk about types of nerves and nerve cells. There are afferent nerves and efferent nerves. Afferent nerves are sensory nerves. These send messages to the brain and the spinal cord. This is how you have sensations of touch, pressure, vibration, limb position, heat, cold, and pain. Efferent nerves, on the other hand, are motor nerves. These send messages from the brain to the target organ or cell. Here's a picture of efferent motor neurons at work. This girl running is using her efferent neurons to move her legs. With afferent nerves, which again are sensory, the cell body is not involved in impulse transmission. With efferent nerves, the cell body of the nerve cell is involved in impulse transmission. We also have several types of nerve fibers involved in pain transmission that we need to discuss today. In the pulp of your tooth, there are two types of basic nerve fibers, type A and type C. Type C fibers are not myelinated, and they create a dull, aching pain. These are the most numerous, and they have very slow nerve conduction. Type A fibers, otherwise known as A delta fibers, are lightly myelinated. Because of this myelination, that gives them a rapid conduction called saltatory conduction, which leaps from node to node of Ranvier. The nerve cell is the structural unit of the nervous system. As we said, it conducts electrical impulses called an action potential. Sometimes we call these nerve potentials or nerve impulses. This picture shows you two types of nerve cells. There's a multipolar neuron and a unipolar neuron, which is the sensory neuron. Let's look at a nerve cell even deeper. This is the structure of a very typical neuron. Notice that it contains an area with the soma or cell body and dendrites. It has a nerve axon that contains a myelin sheath, and then it has an axon terminal which helps it communicate with other nerve cells. One of the most important things about nerve cells, as we mentioned with the A delta fibers, is myelination. Myelin is a sheath created by the Schwann cells. These Schwann cells create a sheath that is interrupted, and this interruption area is called the node of Ranvier. With nerve impulses in a myelinated nerve, this impulse is called saltatory conduction because it jumps from node of Ranvier to node of Ranvier. This helps the nerve impulse move faster. Now let's look at a nerve impulse jumping.
Helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Rampier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Rampier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the
As you could see in that video, the action potential is stimulated by the movement of ions through the bilipid layer membrane of the nerve. And we'll talk about that more later. However, when a nerve fires, we call it an action potential, as you could see. We measure this using a diagram, as you can see here. Threshold is the term that we use to describe the point at which an action potential will actually occur. All of the stimuli, the electrical nerve stimuli that are below the threshold of about -50 or -60 do not result in an action potential. When the threshold of -60 is reached, then the nerve depolarizes, reaches an action potential, and then it repolarizes, which means the membrane goes back to its normal state. Before it gets to the normal resting state, the nerve membrane undergoes a period that we call the refractory period, diagrammed here. During this refractory period, the nerve is unable to respond to any other stimulus, no matter how big that stimulus is. There are two parts of a refractory period. One is an absolute refractory period, during which the magnitude of stimuli will not result in any stimulation whatsoever, and a relative refractory period in which a huge stimulus will result in an action potential. So please be sure you watch that video. I will post the PowerPoint on our Sakai website, and you can watch that again to help you understand neuron anatomy, Schwann cells, the myelin sheath, and action potentials.